Good afternoon. Today we'll be discussing indirect predator effects. I'm Natalie Chavez and these are my colleagues.
I'm Basha Pekarsky.
I'm Bernard Sixer.
And I'm Lauren Sanchez.
And to conduct our experiment, we had help from our mentor, Casillo.
We are using kelp forests to study indirect predator effects. So first I'm going to tell you a little bit about why we should care about kelp forests. Kelp forests are globally distributed. They are found anywhere where there's a temperate coastal region and a rocky reef habitat. Kelp itself has an extremely fast growth rate, and kelp forests are one of the most diverse and productive ecosystems in the world. We don't know exactly what fuels this productivity, but further studies are being conducted to help get to the root of that. Kelp has a great economic value. It is home to many species that are commercially fished, and kelp itself is used in many commercial products, including fertilizer, medication, supplements, ice cream, even your toothpaste. And kelp forests also provide food and a habitat for many important species that are important both ecologically and economically. We will be studying the indirect predator effects in kelp forests, and this can be applied to kelp forest conservation and has wider applications to other conservation studies and behavioral ecology.
So our species of interest is giant kelp, which is also known as Macrocystis pyrifera. Our kelp isopods, which is the Idatea risicata, and our rockfish from the sea bass genus. The kelp isopod is a small invertebrate that grazes on kelp. It only grows about 2 inches long, and one of its main predators is a rockfish. The rockfish can grow up to 22 inches in length, and it's commonly found in the kelp forest.
Alright, so this is our rockfish, and it is a predator. I will be using the rockfish to help explain direct and indirect predator effects. So the rockfish is able to directly affect our prey species, the Idatea, by either consuming it or by intimidating it. Consuming it just means that it's eating it, so it's able to manage the population by the amount of Idatea that it eats. Intimidation would work by when the rockfish is around and the Idatea either sees, smells, or senses movements in the water, it stops grazing and eating and it just goes into hiding mode, so it's not able to gather as much nutrients. Another example of a direct effect would be the effect of the population of the Idatea on the kelp. If the population of the Idatea go unchecked, they just continue grazing the kelp canopy, and the kelp canopy density suffers. An example of an indirect predator effect would be the presence of the fish affecting the density of the kelp. Because the fish affects the Idatea, it indirectly affects the kelp.
Other types of indirect effects, which would be consumption indirect effect and non-consumption indirect effect. When consumption is when it changes the population size of the transmitted species and it's directly affecting the receiving species. But we will look at with non-consumptive indirect effects, where the predator, which is our fish, was not allowed to consume the transmitted species, only intimidate it and change the trait of it, indirectly affecting it, which would affect the receiver species, which was our kelp.
So in order to do these studies, we had to isolate different variables and make sure that the fish weren't able to consume the Idatea. So we had 4 different treatments with both the Idatea and the fish present with the kelp. Just the Idatea allowed to graze the kelp, just the fish with the kelp, and then the kelp alone. So our figures show these dashed boxes around our treatments, and those were to represent our containers. We had clear plastic containers that had mesh covers on them that allowed the Idatea to see and sense the fish, to smell the fish, and they were even moved around by the fish in the tanks. But the fish weren't allowed to get into the containers, so they effectively weren't able to consume the Idatea.
Our controls are important because without our controls, we would have no point of reference. We need them because if all we have was a dependent variable, which was our egg, all we would have is our egg, the amount our Idatea would consume in the presence of a fish. But we need to know how much the Idatea will consume without the presence of a fish, so to compare to see if there's any change. We also have a kelp loan just to account for any natural decay that may occur.
We ran 2 trials of our experiment. The first was a 48-hour period, and the second was for a 24-hour period. Each of our trials, we used 40 containers, which you can see. You can see that this is a container floating in one of our tanks. The arrow is pointing to mesh here, and there are Idatea and kelp inside this container, which kind of effectively shows that our Idatea and kelp were kept separate from the predator, our fish right here, but they could see and sense the predator in other ways.
So throughout our experiment, the information that we had to collect was around the kelp. We focused on the mass of the kelp at the beginning of the experiment and at the end of the experiment to monitor the change, the mass of the Idatea that were within the container, the number of Idatea that were contained, and we made sure to know any abnormalities that may have occurred throughout the experiment.
So we can conclude that if the kelp consumed with the presence of the fish equals the kelp consumed with the absence of the fish, then we have no indirect effect. However, if the kelp consumed with the presence of the fish does not equal the kelp consumed with the absence of the fish, then we can conclude that there is an indirect effect between our predator and our kelp.
In this graph, we have our x-axis, which is our treatments. Our treatments were when the fish was absent, then when our fish was absent, but no Idatea present either. And then when our fish is present, then when our fish is present, but without any Idatea. And our y-axis is when the change of the kelp biomass in milligrams per day. As you can see, when the fish was absent, the Idatea would consume more. So the heavier change in the kelp biomass. And when the fish was present, there was a smaller change in the kelp biomass. And in the next graph, we have our treatments again on the x-axis, which is only our fish absence and our fish present. On our y-axis, we have our kelp change into and the kelp was changed put into the amount of biomass with the Idatea. So to see how much the fish, when the fish was absent, how much the Idatea would consume was about 38% of their whole biomass. When the fish was present, they would consume about 35%. So it was a relatively small change.
So in summary, our data showed evidence that there was a weak non-consumptive effect of the fish on the kelp.
There have been other larger studies on the indirect predator effects in kelp forests. One such study was published by Davenport and Anderson in 2007, and they did find evidence that there was strong indirect effects in the kelp forests. But based on our results, this suggests that the consumptive indirect effects were likely more of the cause as opposed to the non-consumptive indirect effects. We would like to thank the Condor Effects program and everyone who organized it. Thank you to Wendy and Dean and Jens for helping us with our PowerPoint, and Christy for everything, for teaching us and giving us advice and life advice, not just about this project. And thanks to everyone else who participated. We had a great time.